Welcome to today's webinar about microbiome and eosinophilic esophagitis, featuring Amiko Uchida. My name is Jennifer Roeder. I'm a communications consultant for APFED, and I'll be moderating today's webinar. We are pleased to have Dr. Uchida join us today. She's a gastroenterologist at the University of Utah School of Medicine, the co-director of the University of Utah Multidisciplinary Eosinophilic Gastrointestinal Disease Clinic, and is a member of the Consortium for Eosinophilic Gastrointestinal Disease Researchers, also known as SEGR. She specializes in caring for and conducting research focused on patients with immune disorders of the GI tract, such as eosinophilic GI diseases or EGIDs, and inflammatory bowel disease, also known as IBD. Welcome, Dr. Uchida.
Thank you so much, Jennifer. I'm really excited to be here, and thanks everyone for joining now and in the future as well. So that was a really lovely and kind introduction. I appreciate that. I hope to take you all on a journey today where perhaps more questions than answers are will be swirling around in your minds by the end of this. But I hope that that will fuel kind of the next generation of questions and experiments that we're going to explore within EOE, or eosinophilic esophagitis, and its relationship with the microbiome. All right, so here are my disclosures. And now kind of diving in now to talk about the objectives and a little bit of an overview of where we're going to go today. What I wanted to do is really kind of start off, I understand this group, you probably have some familiarity with what eosinophilic esophagitis is, or EOE is, but I want to make sure that we're all on the same playing ground and playing field for how we move forward through the talk. So we're going to review the disease as well as some of the disease processes. And then we're going to move on to thinking about some things like environmental factors, like what are those and how might they relate to EOE? And then we'll move on to, of course, one of the big portions of this talk is defining what the microbiome is and the factors that influence it. And then, of course, how it relates to EOE, or how we might be able to start thinking about that. Okay, so just kind of stepping back and thinking, like what are eosinophilic GI diseases, or EGIDs? Maybe you've heard them called before. And I guess in its most basic form, we can think about them as being increased eosinophils in the GI tract that are, above some sort of baseline level, depending upon the area. And they're either causing pathology or disease, or they're there as a sequela of that. And so it seems kind of simple, too many eosinophils, but the truth is that it can get a little complicated depending upon where we are in the GI tract, and defining what's normal and abnormal, as well as, what are the eosinophils doing there, and what do these sort of things, what are the functions that they have there, as well as the other cells that are thought to cause inflammation, which we're also going to briefly touch on. So this is kind of like high level, big picture. We're talking about inflammatory diseases of the GI tract. And then if we hone in a little bit more, because we're going to really be spending the majority in all of this talk really focusing on EOE, or eosinophilic esophagitis, focusing just on the esophagus, though recognizing that you can have too many eosinophils, if you will, in other places in the GI tract as well. So when I think about this disease and talk with my patients about it and others, some of the things that I like to point out are that I think of EOE as a chronic disease, okay? And we think that it's largely food driven, okay? And it is an allergic disease that affects the esophagus. And the reason I like to point out the chronic component is because, unlike some other diseases or medical problems where perhaps you get a treatment for it that lasts maybe a week or a few weeks or something like that, and then it kind of goes away, inflammatory diseases of the GI tract, and EOE in particular, are not ones that just sort of go away on their own. They require some sort of treatment and some maintenance therapy over time as well. And so things that our patients will experience when they have this disease, we're going to review in a little bit more depth, but sort of big picture, a lot of patients have trouble swallowing, getting food down, and sometimes that food gets stuck in the esophagus. And I found this photo on Google Images, which from the citation with shown there, but I find it illustrates things within EOE, I think really well. This person is holding, or this drawing is holding a piece of chicken, having difficulty getting that meat down. Meat can be a really challenging food to navigate through the esophagus. And you can see that microscopically, there's inflammation there, specifically with eosinophils, which it's worth noting that at baseline in a healthy or normal esophagus, there really are not eosinophils there. So that means that when we do see them, it becomes a little bit more clear and easier, if you will, to say, okay, that's not normal when we see them there, particularly when we see them in high abundance, which is what we end up doing when we take biopsy samples and looking at them under the microscope. We end up quantifying them, which again, many of you are probably familiar with. So I will also kind of point out that some of these symptoms that we're reviewing here can be different across the age spectrum, and that's what we're going to highlight here. Okay, so patients with EOE, when they're really young, our pediatric patients, for example, oftentimes they are not having specific symptoms saying, hey, I have a hard time swallowing. That would be a bit unusual. Oftentimes patients that are under the age of 5, for example, you might be picking up on things like just trouble, difficulty getting them to eat, difficulty with feeding episodes. We use the term failure to thrive kind of as an umbrella term about the child or the patient is just, it's not doing well. Maybe it surrounds feeding or energy levels and whatnot. And oftentimes you can see patients gagging on foods, particularly solid foods. As they get a little bit older, some of that localization to the GI tract, more with trouble swallowing, can start to be seen, but you can still have some, less targeted symptoms like vomiting or abdominal pain. Some people even talk about bloating. And it's really not until patients get a little bit older into their, teenage or later teenage years and high school years, where that localization ability to come to the esophagus and say, hey, I'm having trouble swallowing. When I eat this food, that's hard to go down and feels like it gets stuck. That doesn't usually happen until these later ages. Excuse me. And I've also noted on there this term strictures, and we're going to revisit a lot of these terms again. So I'll kind of like put a little plug out there for them and circle back as well. And so what I mean when we say strictures, that I'm talking about scarring that can happen in the esophagus. The other thing that's probably just worth noting from this clinical perspective, and maybe goes back to the chicken that the cartoon was showing on the prior page, is that it's not necessarily that the food that a patient or a person is eating at that time is their food allergen or their trigger. Most of the time, it's actually that is a food that requires more coordination of the esophagus to navigate getting it down this food tube into the stomach. And those foods can be kind of stickier, if you will. And so that's something I commonly run into and talk with patients about when we're talking about symptoms here. So I also want to kind of highlight briefly, at least, some reasons why I think we should care about EOE and about this disease. So when we look at populations actually across the world, we actually find that there is an increasing incidence of the disease. So incidence means that basically more and more people are being newly diagnosed with the disease. And so you can see this graph. This graph is actually several years old, but is showing in these various populations, which are colored differently here from different cohort populations, that we're seeing that the incidence, the new diagnoses are going up. And because this is not a life-limiting disease that we know of, at least at this time, the prevalence or the abundance, the number of people that end up having the disease is increasing, right? Because we've got a faucet that's turned on and we're diagnosing more people. And then the total pool of people is therefore also increasing. Another reason I think this disease is really important is that it actually can affect all ages, all sexes, and all races of patients. And so this is something we need to be thinking about for everybody. The proportions of those various factors may be different between groups, but again, it really is something that we need to think about for everyone. The other thing that if you are a patient or a caregiver for somebody with EOE, you probably know that this is a costly disease. Some of these analyses are years old, but there's a lot of healthcare touches that are required with this disease and the maintenance. And we try and, of course, prevent things that land people in hospitals and emergency departments, like if food really gets stuck. But this is a disease that requires a lot of healthcare touches. And we have only one FDA-approved therapy so far. So many of the treatments that we do for EOE are not FDA approved. And we're going to briefly highlight those. Then the last thing that I'll mention here, as I kind of briefly introduced on a prior slide, was this idea of scarring of the esophagus. And these are some of the more long-term issues that can happen with patients who have EOE, that you can get scarring, fibrosis, or stricturing. Those are all very similar terms that you might hear people use, either your doctors or yourself use, when referring to some of the problems that we encounter with EOE. So we know that the incidence is increasing, but let's think now a little bit about why. So I have here a little bit of speculation about why the frequency might be increasing. And I wanted to highlight this because I think it brings us to thinking about reasons that the microbiome might be relevant. So one of these things, and also this is not an exhaustive list, one of those things that I've mentioned here or put up here is the hygiene hypothesis. And there's a lot of different ways of sort of interpreting the hygiene hypothesis. You may have heard of this before. Perhaps it's been thought of or spoken about in that we live in too clean of an environment, if you will, in these westernized societies. We don't have the same or the diversity of microbes that we were exposed to before. And this ends up leading to a either untrained or bored, if you will, immune system that then ends up developing reactions against things that are not harmful, right? Like food, like pollens, aero allergens, those sorts of things. So that's kind of a big picture view. And we think about this from the perspective of other immune diseases too, like autoimmune diseases, with this same kind of umbrella idea, particularly within types of microbes called parasites that might be more relevant for allergy. Parasites are quite absent from westernized societies like the United States. And so is it that, our immune system is sort of lacking that exposure and training of our immune system? It's possible. So the next thing that I have listed on here is, in air quotes on purpose, called microbial dysbiosis. And this is a term, again, you're going to see on a couple of different slides that we're going to come back and define and maybe redefine. And I say that because we don't totally know we really don't know what a normal microbe composition is. And we'll talk about that a little bit more later as well. But I think what a lot of people mean when we say microbial dysbiosis, we're talking about microbes that then end up getting a perturbation or a change. There's a change in what was there, and there's a lot of different ways we can impact that change. But now figuring out, is that change bad, good, or neutral? Those are the sorts of things that we don't have the answers to just yet. But this is, again, a term that's a bit of a catch-all that we're going to come back to. So you can see things like maybe diet, maybe exposures like antibiotics or person's lifestyle could be affecting the microbes that are in and on us. And then the last thing that I've highlighted here is actually very similar to the above. It's asking us to think about our environment and how our environment might be impacting not just us as humans and our cells, but also the microbes that live in and on us. Okay, so again, factors and changes like food production and how those can be modified, chemical additives and processing, and even the air that we breathe with pollutants. So these are just questions that circulate around and come back to this idea of our environment and how our environment may be impacting disease processes, okay? So this next slide, not intended to be overwhelming. There's a lot of cool pictures that are kind of interactions that are here, but I really like this this slide, and we're going to simplify it and hone in on a couple things that I think are important for this talk. But the bigger question is sort of like, how might these external things like diet and microbes be affecting EOE? And so this is actually a figure from a paper that was published about 5 years ago by some EOE disease researchers. And they're trying to distill down, like, what is it that we know about the disease process in EOE? And so here, if we think about these upstream signals or factors or contributors, right? These upstream things here far to the left, we see different food and dietary inputs. And you also see microbes input here as this higher upstream signal. And now what might they be contributing to this downstream inflammation or inflammatory process is still yet to be determined. And we're going to go through some of the data today that has been done. But this is, again, kind of this big picture idea to get us start thinking about how these things might be interacting. Okay. So this slide is intended to really sort of tie together some of the things that we've talked about so far. We know now what eosinophilic esophagitis is in some forms. We have these various inputs, things like food and aero allergens. We didn't spend time really talking about how there may be actually host or patient genetic, your DNA code differences that might predispose you to developing EOE. But that's certainly thought to be a potential factor feeding into and or driving this disease. And then we've got environmental factors. And in truth, we could put food under environmental factors as well, right? These are external things that might be contributing. And then we've got our esophagus here that ends up getting inflamed. And this is a picture from one of my patients from one of their scopes, which we use to help monitor and diagnose the disease. These are some of the cell types, of course, the eosinophil, which it's named for, that we end up finding coming in on those biopsies. When I mentioned we take samples and we look under the microscope and see, hey, who's here? What's going on? We see a lot of inflammation. So I know this picture is really small, but those pink cells really just shouldn't be there in a normal esophagus. And over time, that whole process can really lead to fibrosis and scarring and making it difficult for the esophagus to contract as it normally would. All right, so I want to focus a little bit more now on reviewing a little bit more about these environmental factors. So we're going to spend a little bit of time thinking about how that might actually be playing into EOE. So there are certain environmental factors that have been studied in mostly population level research studies, okay? And so what are those environmental factors? So I've listed a couple of these here that could be contributing. And so when we think about antibiotics, right, you're prescribed antibiotics for an infection. So some microbe has become a problem or a disease problem for an individual. And so we give an antibiotic in efforts to try and alleviate that microbe. But in the process, antibiotics are not perfect, and they're going to remove other or decrease other populations and communities as well. And similarly with an infection, you get one or a couple of microbes that are kind of taking dominance and causing a problem. And that ends up shaping and changing not just the bugs that are there, but also our immune system, right? It gets activated as well. And then, of course, the things that are feeding and nourishing us are absolutely the things that are changing and nourishing our microbes as well. And so when we think about these various environmental factors, it begs the question of how might this actually feed into and contribute to this idea, again, of microbial changes, dysbiosis. There's this change that's happening. Is this a harmful change? Excuse me. And so some of this work, at least particularly within looking at early life exposures, things like getting antibiotics early in life, has been done by Dr. Elizabeth Jensen, who some of you may have heard speak at these events before. I believe APFED will be able to connect you with an earlier talk on her work as well. And in one of her studies, she actually investigated if patients who had an earlier life, so young in life exposure to antibiotics, did that end up portraying or showing an increased risk of developing EOE over time? And she's done this with other environmental factors as well, but we'll focus on that. And she actually did. She found that there was quite a high increased risk of developing later eosinophilic esophagitis when you had antibiotic exposure at a young age. And so in efforts to sort of study this further, because this was in a smaller population, work that I did with a collaborator in Sweden actually did this at a nationwide population level to take biopsies from patients who we have a cohort where we know that they have EOE, and to follow them and determine, did you get exposed to antibiotics or infections earlier in life? And so when we did that, we actually did find that infections are associated with a twofold increased risk for developing EOE. And when we looked specifically at antibiotics as well, we said, okay, here are patients with EOE, and we look back in their history and say, did you have exposure to an antibiotic? And we found that those patients also displayed an increased risk of developing EOE later who had had an antibiotic exposure earlier on. And so the idea around this, and that I was hoping to plant this seed, is that these environmental exposures sort of raise questions about what protective effects actually that microbes may have, particularly within the data of antibiotics, right? There might be some select pathogens that are problematic, but there also are these other ways that we can think about microbes and bugs that are on us and how they might actually be protective. Because when we give antibiotics, for example, and get rid of some of them, how is that leading or being associated, I should say, with a later risk of developing EOE? So that's a little bit of the segue into what are we talking about when we talk about the microbiome. So I'd like to spend a little bit of time on this to dive into this a little bit more. So in general, what we're talking about are things like bacteria. We're talking about fungi, as well as viruses and parasites. There actually are others as well, but that we're not really going to focus on or mention here. And when we think about these collectively, there are trillions of different microscopic bugs that live in, on, and among us. They're sharing in our every moments, in our meals, and in our activities. And what I've hopefully tried to do over the last several slides is start to plant the seed that not all microbes are bad, right? Such as in an example, such as the hygiene hypothesis, right? Where maybe some are important for training our immune systems and our bodies, and maybe some are important for helping us have this balance and normal, this term homeostasis that we like to use, where we're kind of at a balanced state of health. And I think as a scientific community and as a culture, really, we're really only beginning to understand and appreciate that these masses, right, trillions and trillions of microbes that we live with are, for the most part, symbiotic with us, right? That we're, or at least there's this term commensalism, right? Where we're living together in a sort of harmony, if you will. And of course, there can be, bad or pathogenic disease-causing incidences that happen as well, and we have treatments for those. But we really want to think about this as health and disease, and how this is a constant balance with both us as humans as well as the bugs that live on us, okay? So the other challenge that I want to put out there for our brains to wrap our heads around is the idea of what's shown here. And you're not expected to be able to read this text, so please don't. But the idea with this was actually a really nice review snapshot that was put together by one of my mentors, June Round, and friends, Jen Hill, in the lab, with the idea that really microbes in a host or in a person, right, can actually have impacts and affect the function or the physiology throughout the body. So even if the microbe is, say, in the intestine of a patient, there have been research studies that have shown that there can be effects in places like the brain, and so our nervous system, as well as our muscular system, our cardiovascular system, that it doesn't necessarily have to portend to the microbe that is right next to a certain area. And this is an idea that us as researchers and scientists are really only beginning to start to explore and understand truthfully. And those effects, how can a microbe in the intestine have effects on the brain, right? This is a really big, important question. And there are various ways that we think those things can happen. So microbes can actually help us digest things that we eat, and they can help us, they can actually produce factors, metabolites that can actually circulate and travel to distant sites and have effects. And some of those effects can be beneficial. And we also recognize that some microbes can actually be detrimental, right? And it all kind of depends on the context. And so there's a lot of nuance with this and within the microbiome research, okay? So again, perhaps maybe more questions than answers at this point, but I hope we've got you thinking. What I'd like to transition to now is thinking about how do our current, coming back to EOE, how might some of the things that we do within EOE actually impact or have the potential to impact the microbiome? And so Some of you may are hopefully familiar with some various treatment forms for EOE, but I was hoping to focus on a little bit with our thinking, and some of the data that I show you is a little bit more focused on diet, because that's kind of a really obvious one where we know from diseases outside of EOE, and outside even of the GI tract diseases, that when we change our diet, that certainly does affect the communities and the populations of microbes in our intestines and in other places. And so it's so interesting that we have this disease, EOE, where actually one of the mainstay treatments is a diet modification. And we understandably think of that about removing that food. We think food trigger or food allergen. But I also want to challenge us to think about what effects that might be having downstream on the microbes as well. Okay, so here's some pictures of the types and categories of foods that we generally think of as being triggers for EOE, and again, getting us to think about how they might be when we eliminate things like dairy, wheat, eggs, right? These are generally big staples in a westernized diet, if you will. And, other foods like soy, nuts, fish and shellfish, etc. These are really common to a lot of diverse cultural diets as well. And so when we're eliminating these, what are some of the effects that could be seen within the microbiome? And interestingly, there was a study done on patients not with EOE, but who were put on a gluten-free diet. And even within a few weeks, they were able to measure in the stool of patients before and after that dietary change that there were changes and differences within the microbiome. Now, whether or not, again, those are beneficial or neutral, for example, we don't always know, and that will depend probably upon each disease and disease state. But beginning to think about things in that way with an EOE, I think is really exciting and interesting. Excuse me. So there's a lot of different things that we can explore, and I feel like we've got to get busy and starting to investigate this. So this brings us to really asking this question, like, is the microbiome important in EOE? And we're really in the beginning stages of gathering data on this. And so what we're going to go through now are some of the work that's been done so far within EOE specifically and the microbiome. And then hopefully this review is going to help inspire us to come up with other questions as well. So to give you a little bit of a preview, I'm going to walk you through they can be kind of complicated graphs and bits of data that has been published over the last several years, but we're going to walk through it and take it step by step. And the other thing that I'll kind of navigate you through is that people have taken samples from various sites and disease states. And so I want to help orient you to, this sample was taken from this site. And so you'll see that here, we're going to do a little bit of a breakdown for defining these different sites where things have been taken from. So far, most of these samples within looking at the microbiome and an EOE have been taken from places like the oral cavity, the mouth, also the esophagus, of course. And then there have been a couple of studies that have actually looked at stool as well. We're really just kind of beginning to explore there. And there's various ways that investigators have collected these samples, which I've shown here. One of which maybe you've heard of as the string test. That's going to be some of the first data we look at. And then they go on and do what's called sequencing, and I'm going to use the term sequencing and identification. I want you to kind of think of those as very similar things. And so far, all of the forms of identification of what the microbes are have been done using a technique called 16S sequencing. I wouldn't expect you to have heard that before. But this is a very common and well used and described way of sequencing. It kind of provides like an X-ray image, so of what's going on, but they're recognizing that there actually are other techniques that can be used to define and identify what microbes are that have different and higher resolution, and those things haven't yet been done within EOE. So a lot of what we're going to go through here now moving forward is you'll see whatever site I'll try and, for a given study, I'll try and cue you in with one of these organs or tissues. And then we're going to be looking at relationships between diseases and disease states, as well as microbes, okay? So if we start back, this was a study done by Harris and their group, and they were looking here at the esophagus. And one of the kind of big picture first steps that you can do within a microbiome study is to just sort of take a step back and say, hey, is the amount, like the load, is how we call it, bacterial load, is the amount of bacteria different between disease state and not disease state? So you see EOE here on the right, and patients who didn't have EOE here on the left. And each dot or each square here, and you'll see this throughout, really represents a given patient or patient sample. And what this group found when they investigated this, again within the esophagus, is that they found that the bacterial load, the abundance there, was higher in patients who have EOE. And so then as a next study and as a next step that they went on to look at is they said, okay, well, there's more bacteria. What about the components, of the bacteria that are there? What's the diversity like? This is another really common thing that is done within microbiome research, is to say, what is the community like and how diverse is it? Within biology in general, we kind of think of diversity as being an important and helpful or beneficial thing, right? It gives you more options for perturbations that might come in or stressors that might come in. If you've got more diversity, you might be able to rebound and be more flexible, if you will. And so there are various ways of measuring diversity, and this is just one method that they've done here. And in this study, they didn't end up finding that when they looked at what are the components, like the diversity of communities within the microbes in patients with and without EOE, they didn't end up finding big differences here. But this is, again, another high-level way of examining complex systems like the microbiome. So this is work from a different group done around a similar time. And this is like a really cool and colorful image, but it could be a little confusing. So we're going to break it down, and things that I want you to focus on. So this again is looking at in the esophageal microbiome, okay? And this group really felt like from their work that they were able to say, the microbiome in the esophagus really appears to be distinct. And one of the reasons that they felt that way is if you focus on the left-hand side of the screen here of this circle, this cladogram, these are patients who are controls, and on the right, and this is shown in red, these are patients who had active disease or active EOE. And what you probably cannot read are these different groups they're called phyla, these different groups of bacteria as a way of us kind of dissecting out potential differences. And what they found is that this group or phyla called firmicutes tended to be higher in patients who had who did not have EOE, who are control. So I'm showing you that here in green. And then patients with active EOE tended to have more of the bacteria that are found under this overarching umbrella group called proteobacteria. So those were higher in EOE. So that seemed to be distinct between these two groups. The other thing that this study did is they said, well, hey, what if we look also in the mouth, because the mouth is right before the esophagus and we're kind of constantly swallowing. Are the microbes that we see in the mouth reflective, or could they be a surrogate? Are they similar enough to the esophagus that we might be able to determine some findings by just looking in the mouth? And so this is another way that they've shown that this is another way that we can display really complex systems as well. So what I want you to do is sort of focus here on the left in this box that I've outlined in red. So that these samples represent samples taken from the esophagus, and all the little colors that you're seeing in the pattern, these are the different microbes from different samples from different patients. And then similarly on the right-hand side, this is from the mouth. And so if you sort of take a step back and you just look at these two halves of this square, we're looking for similar patterns, and there are statistical and more sophisticated tests that can be done. But what they ended up concluding is that they are similar, the microbes that are found within the mouth and the esophagus, but there are distinct and notable differences here. And I think you can begin to appreciate some of that, where you see patterns here in the esophagus on the left that are just not present on the right. And so while interesting, it seems like that they are similar, it seems as though the mouth and the esophagus are two separate places. Then we need to think about them perhaps separately in regards to the microbiome at least. Okay, more work that's been done. So we were talking at, high level, big groups that encompass lots and lots of different types of bacteria when we talked about firmicutes and proteobacteria. What if we hone in a little bit? So some people ended up coming down and saying, okay, well, what if we look at this smaller group of bacteria called Haemophilus, or any other types? Are there any differences that we find in patients who have disease or non-disease? And this group looked at, again, patients without EOE, and then they actually separated based off of if they were treated or not treated, and ended up finding within their data set that Haemophilus, which is a type of bacteria, seems to be increased in patients who had untreated EOE. And similarly, another group used slightly different patient population, but again looked in the esophagus and found that a different type of bacteria called Streptococcus, or strep, as you've maybe heard, which is still an overarching group. If you look here down at the base of this, within this active EOE group, we see a big pink dot, and that's identifying this bacterial group called Streptococcus. They found that was actually abundant in active EOE. And data that I'm not showing you here, they also were able to find Haemophilus was increased in active EOE as well. So it's nice, we're seeing some repetition with different patients and different investigators, but we're still seeing that some things are shared and repeated. So that's always nice within research. So now we've been focusing a lot on the esophagus, which makes a lot of sense because this is the site of the disease, but really cool to step away and say, hey, these investigators a couple of years ago actually looked at the stool where a lot of the microbes are located, right? And they looked at stool of patients. These were pediatric patients actually, who have EOE and those who do not. And this is a different way of looking at microbiome data as basically identifying different populations. And so what we're showing here, this is a key over here on the left of different names and groups of bacteria. Some of these, again, should sound a little familiar, firmicutes and Bacteroides. So if you focus here on the left, we've got 12 patients who didn't have EOE, and we've got 12 patients on the right who did. And what we're looking for, again, at this level is just pattern recognition, right? Do these look similar or different? Well, there's similar colors. We're seeing the groups might be, the types are similar between them, but maybe the proportions are a little bit different. And so then they went on to do some more studies to say, all right, well, what other differences can we sort of dissect out within this, within these stool samples of our patients? And so we come back again to that idea of diversity, right? This is a way, it's called the Shannon Index, as a way of looking at how diverse are the microbes within a control patient or within an EOE patient. And this group ended up finding that there what seemed to be decreased diversity within patients who have EOE. Again, this is within their stool exams. And then honing in on this a little bit further, so we saw these big phyla level differences signaling that there might be a difference. And so if we can kind of hone in a little bit more and get a little bit more specific with what groups of bacteria might be different, they honed in on this group of bacteria called Clostridia or Clostridialis, and ended up finding, when you compare patients who don't have EOE to those who do in this small study, that it seemed as though EOE patients had fewer or had less Clostridia within their stool. So what, if we stop and say, all right, we've reviewed a bunch of data, what does some of this mean? Like, what are some of the takeaways? And so for me, so far, it seems that there are some studies that have indicated that there are some differences both in the esophagus as well as in the stool. And these are, for the most part, have been done in small studies, right? In smaller groups of patients. And other questions that are still remaining are, does this replicate when we add more patients and look at a more diverse patient population? And what do we think about the function of this? Like, just because it's decreased, does that mean it's good, bad, or neutral? And those are questions that we still have yet to answer. So this is the next bit of data that I wanted to review with you is data from Seeger, the group that Jen actually mentioned at the beginning. So we're a big group of GI researchers across the country and even abroad. And we're really focused on studying patients who have eosinophilic GI diseases. And what I'm showing you here, you're not meant to take all of this in, but I like this illustration because this group, we took together and said, okay, there's been a lot of these little differences in smaller studies. What can we do as a big group to help move things forward and sort of verify and at a larger scale some of these findings? And so for this paper, they actually also looked at patients who had gastritis. So, but we're not going to focus on that, which is over here on the right-hand side. But what they did is they standardized the approach of increasing the number of patients that we're looking at and looking both in the esophagus as well as stool, actually, to say, can we replicate some of these things? And so some of the data that we're going to review today, here's some that most of it was done within the esophagus. And they found that sort of overall, it seems like the microbial communities in the EOE esophagus seemed to be changed or altered a bit. But when it came down to coming to statistically significant differences in things that we've talked about before, like the abundance or the bacterial load, if you will, or the diversity of various communities within the esophagus of these different groups, we've got control patients and patients who have active or inactive EOE. They really didn't see striking differences or big significant differences in either of those metrics here. And so another piece of data that they looked at, and this is similar to that colored bar chart that we looked at a couple of slides ago, where we break down the different bacterial groups and communities based off of color. If we look again at controls, active or inactive EOE with these 3 bars here, you can see that the colors are fairly similar between the groups. The proportions do seem to differ a bit. And so they were able to point out some of that. So for example, with inactive, you see that this brown chunk of the bar is more than in inactive or healthy control. And that reciprocal difference is seen within the red. And so we think about, oh, okay, well, that's interesting looking at some of these species here. Strep is what's noted here, and another Prevotella, these groups and communities. So we're starting to describe the landscape a little bit more in a bigger population, finding some of these differences. They then went on to dissect things out and say, well, what about the types of treatment? If you're on a particular type of treatment, does that end up making a big or significant difference between amongst your microbes that are in your esophagus? And so these listed here are some of the various types of treatments. So we've got proton pump inhibitors, steroids, and diet, and we've got mixtures of those different approaches as well. Overall, again, you can see that the landscape, the colors look similar, but those proportions vary a little bit between groups. And overall, though, when they looked at this, they didn't end up seeing any statistically significant community differences between these various treatment groups. And when you have so many groups, it becomes challenging to find statistically significant differences. The differences have to be really big in order for you to see them when you have lots of different things to compare. And data that I'm not showing you here, I did mention that they did collect stool samples from some patients, and some of the comparisons that they ran there were just to look for big, broad picture, again, differences. Are there big differences between the stool of patients with EOE and without? And that they didn't end up finding big differences in this larger study. So the last piece of data that I wanted to end on, I thought was really interesting. This actually wasn't done within a population of EOE patients, but this was done to investigate how diet can actually change the esophageal microbiome, which is I think quite novel and was surprising. And so these were patients who were eating a low fiber diet or a high fiber diet, and they were sampling the esophagus as shown here. And then looking again at these big picture phyllo level changes, what are the communities like? And you see a more red in the high fiber diet, compared to the low fiber diet, for example, among other differences. And they did some other analyses as well. And this graph, again, is not meant for you to be able to read the teeny tiny print, but showing what's shown here on this axis is fiber intake and how that tracks with the relative abundance of this group of bacteria, firmicutes, that word that kind of keeps coming back to us. And how it sort of inversely is associated, a high fiber diet is inversely associated with a different group, that proteobacteria, right? We saw more proteobacteria in inactive EOE. And so one of the things we can conclude from this paper, high fiber diets were actually associated with higher esophageal abundance of this group of bacteria called firmicutes. And again, this was outside of EOE, but I think it's really interesting that we can change our diet and end up having impacts on the microbes that are in and on us and among us. Okay, so let's summarize things with some take-home points here because we're about to time and we want to save time for questions. So I hope that we've been able to, that you've been able to take away that there are various environmental factors listed here, things like infections, antibiotics, various changes that can happen within our body or from the external environment that have been associated with EOE and may impact the microbiome as well, based off of what we know from other diseases. I hope you've been able to appreciate that the microbiome is actually very complex. And the big thing that I'm really hoping to take away, that people take away from this, is really thinking about the microbiome as a not just complex, but also beneficial community, right, of organisms, that they can be beneficial and some microbes certainly could be harmful, but we want to think about them in this more diverse realm. Researchers have also found some differences or changes in various sampled sites from patients with EOE within the microbiome, right? We did see some differences in smaller studies, and those weren't always replicated at larger studies. But there's still so much for us to learn about how a given controlled intervention, such as diet, might actually change a certain thing within an individual or group of individuals with their microbes. And that's really what I'm pointing out here is, again, specifically how can our treatments end up affecting microbes and the downstream function of those microbes interacting with patients with EOE. So I think I'll pause here, and I just again want to thank the organization and all of you for joining today. We have, I think, about 15 minutes to review questions, 10 to 15 minutes to review questions.
Wonderful. Well, thank you so much for all the wealth of data that you just shared. We appreciate it.
Of course.
Wonderful. Well, if you'd like, you're welcome to stop sharing your screen while we do the questions. All righty. So we've got some good questions coming in here today. So the first question is there a relationship between higher bacteria and more hygiene?
That's a great question. I'm going to assume sort of within EOE, but I'll try and answer both ways because it could be, elsewhere. So I think the answer certainly within EOE is that we don't know. This is though where the hygiene hypothesis comes from. Again, we apply this to other immune diseases though, like autoimmune diseases. So finding that direct correlation can be tough because it's not necessarily that we have fewer bugs, if you will, right? We're still on the order of trillions, but perhaps the type of microbes that we are encountering, right? So if you think about the types of microbes that we encounter in Salt Lake City, where I am, or within these westernized communities, we're not seeing a lot of parasites, for example, right? And by parasites, I'm talking about things like worms that can live, in and among us. This is something that we evolved, though, to combat. And when we think about allergy processes in particular, the cells like eosinophils, they're super important for getting rid of and controlling things like parasites, right? And so this idea that microbes, the component types rather than number are different between populations that still have parasites, where we don't see these types of diseases with any sort of frequency like we do here, allergic diseases, autoimmune diseases. This is some of this indirect data that helps fuel us and makes us wonder about how these microbes in quality might be affecting training of us and our immune system, rather than just a sheer number. That's a little bit of an indirect way, but I hope that kind of answers. The answer is we don't know exactly.
Well, we appreciate you giving more insight to it. Another question that's very specific to EOE is high fiber diet good or bad for people with EOE?
It's a great question that we don't know the answer to. And that's also, remember that last study that I showed, it's something I wondered about as well. And I thought that, when I try and we as scientists try and, read broadly as well. And how can we take what we know in other diseases and maybe apply it to a disease that we're focused on? I thought that that was a really interesting study that showed that we can make changes in the microbes in the esophagus, but what does that translate to for my patient in front of me? So we have to study this, and this is something that I'm very interested in doing, and a part of my growing program here at the University of Utah is things like fiber and diet and how these make impacts and changes on microbes. So I don't know if it's good or bad yet, but we should figure it out together.
We've got a number of questions coming in about pre or probiotics. Is there any of the research that you've seen that has information about a correlation between EO and EO in them? For example, some of the questions coming in, should EOE patients be taking pre or probiotics? And what can it be helpful in treating EOE? What role does a probiotic play? So it's coming in from multiple angles, but you've gotten people thinking.
Yes. Okay. I'm really glad about this. And remember, I opened by saying we're going to have more questions than answers after this talk, which I think we're kind of getting there. So I will say pre and probiotics, not just within EOE, in general, we are not at the granular level yet of our understanding for saying we can apply that these are beneficial and have been shown in research studies to affect diseases, right? So even for diseases outside of EOE. So there haven't been any in EOE. That one is kind of a quick answer. So I don't know, and I can't tell you that it would be helpful. And truthfully, from a numbers perspective, the way things have been done so far within other pre and probiotic research studies to look for benefit for XY disease, there have not been very many studies that have actually shown that pre and probiotics are, can be broadly applicable to, inflammatory bowel disease, for example, another disease that I study, which involves the GI tract. There have been some studies with very specific types of probiotics that have been shown to prevent some outcomes, like within inflammatory bowel disease, things like pouchitis or inflammation if these patients have had surgery. But I just mean to kind of convey not necessarily those level of details, but that broadly speaking, we have got a long ways to go at having a standardized approach for our microbial makeup, right, of that probiotic, and or what prescription we might put together or concoction we put together for saying this type of prebiotic or a cocktail, if you will, is beneficial for this type of disease or this type of patient. So there's a lot of granularity that we and noise, if you will, that needs to be sorted out before I think we can say that yes, this is going to be helpful for that. Is it likely to harm you by taking prebiotics or eating fresh fruits, vegetables, which are amazing, fermented foods, which could be amazing prebiotics? It's probably unlikely to harm you. But there's a lot of regulation that is just not known right now. And as you probably know, these are not regulated by the FDA. So the quality and composition, it becomes very, not scientific in that way, because we like to control these things in our research experiments. And so those are important questions that I also have that we don't yet have the answers to.
Well, your comment about control dovetails very nicely with another question that came in.
Great.
People are curious about these studies and are they controlling for active EOE patients who are eating lots and lots of foods versus active EOE patients that are avoiding lots of different foods?
Right. So broadly speaking, mostly no, they're not. That the Seeger study, and these things are hard to do, right? Humans, we're complicated, and doing research on humans is hard, and remembering what you ate is also hard. But the Seeger group was really trying, and some other smaller groups have also tried to take some steps forward into trying to control for some of that, right? They broke it down for, these people were being treated with a proton pump inhibitor. These people were being treated with diet, but what food components, such an important point, what food components were in there or eliminated or not there, that level of granularity of data we don't have. And like you can imagine, doing that for hundreds or more patients and asking them to remember what they've eaten in a controlled way, it's a challenge, but it's a challenge that we're up to, I think. And it sounds like this group is maybe also up to and interested in. So
very much so.
That.
There are way more questions than we're going to get to in the next 3 minutes. So since there are so many things you don't have answers to, what research are you most looking forward to that might provide some answers?
Oh, I really like this question. So I'm biased. So my lab and the expertise here is really within not just, describing what we see in these snapshots, which is really important as a first step, but is really actually honing in on like, what are some specific functional differences, functional mechanisms, like what are the actual benefits or detriments that can be seen, and studying that in a controlled environment. Sometimes we have to use animal models in order to do that because humans are, complicated, and sometimes we can't do all the controlled experiments that we would like. That is something that I am in particular really interested in, is that maybe on this big global scale, when we take snapshots of, 100 patients like the Seeker study, we're not seeing big, obvious, striking differences, but now we need to really mine that and do some more controlled experiments and interventional things where we are going to come in and tease out like mechanism. What is it about this bug that could be helpful or harmful? But kind of thinking of them as, this coin phrase, like bugs as drugs, right? What can we do? And that is really something that our lab has expertise in honing in, not just on these important descriptions that end up happening, but honing in on a community or a type of bug and its actual function with us as humans, and how can we harness that and engineer and see that future further, thinking of bugs as drugs.
Well, I think we're going to all have to stay tuned to your research and see what comes out of it. For those of you who we did not get to your questions today, please know that we have already discussed with Dr. Uchida that we will send her a list of questions, and we will do our best to put some of them on our website so that we can try and get even more questions answered than we have time for today. So I know you have a hard stop today that we need to get you going to. So I'm going to share my screen here and do a few final thank you messages. All righty. Well, thank you again for your wonderful presentation today and all your thoughtful answers to the questions we received, as well as the ones that we didn't get to discuss that you will be answering in the future. So thank you. Thank you also to our education partners for their support of this webinar series. That includes AstraZeneca, Bristol Myers Squibb, GSK, Sanofi, and Regeneron. To continue learning more about eosinophilic disorders, I hope you will register for future webinars. Our next one's on May 9th. It features Dr. Cleon from the National Institutes of Health, who'll be talking about biologics. We also have a panel discussion planned for May 19th during National Eosinophil Awareness Week about diagnostic tools in eosinophilic disorders, featuring Drs. Lerner, Menard Katcher, and Rubenstein. Another great opportunity to not only learn, but also connect with other patients and caregivers who have life impacted by eosinophilic disorders is EOS Connections. It's APFED's annual patient education conference. This year, we're headed out to Denver, Colorado, and Dr. Uchida is going to be there with us. And we're also looking forward to having people join us online. It's going to be a hybrid conference again this year. The main conference is going to be on June 30th and July 1st. And we also have pre-conference activities for those joining us in person in Denver on June 29th. Finally, we also have a podcast series. Our latest podcast episode had a wonderful conversation with a young patient, Ileana, about EUE and unsedated transnasal endoscopy. You might have heard this called TNE. And so she talks about her experiences undergoing that. Also help you to check out our online community on the Inspire Network. You can check that out at apfed.org/connections. It's a great online forum to be able to connect with other people and build community. But Once again, thank you so much to everyone for joining us today, and particularly to Dr. Uchida for her wonderful presentation and all your time. If you have any questions that didn't get answered and you haven't submitted them yet, feel free to shoot us a note at mail@apfed.org. I hope everyone has a great day.
Thank you.